Hello, everyone. This is Miss Peachy again from Biology A, and now we're working on Unit 5, Lesson 8, Making and Breaking Bonds for Energy. So this one is the hard one. When we talk about cellular respiration, the lesson is very hard. Lots of vocabulary here, and I'm just hoping to make this a little easier for you to understand. That's my goal here today. So as you can see from the list of keywords, there's a lot of really, really complex words here. Um, I don't, it's try to just follow what I'm doing because I feel like that will help you understand and kind of simplify things a little bit. So remember, our goal from cellular respiration is to take food, either the food we eat or the food that's made by our own bodies if we're plants, and actually break that down, extract the energy, and make ATP, because ATP is like the gold in living things. It allows it carries high amounts of energy in a very small package, so it can be easily transported around and is just readily available for use. So that's our goal here. So I'm really not going to even go through the vocabulary because I want to go through that as we kind of go through the lesson today. As always, get that note sheet that I have on my website to help you follow along, kind of gives you an outline to work from. All right, so we're going to talk about aerobic respiration. And aerobic respiration is something that requires oxygen. So most organisms do use some form of aerobic respiration. All plants and animals do anyways. And since we're kind of modeling in plants and animals, we'll talk about aerobic respiration. Aerobic respiration takes place in two different areas, in the cytoplasm and in the mitochondria itself. So the three parts of aerobic respiration are glycolysis, the Krebs cycle, and the electron transport chain. The first step of aerobic respiration takes place in here we go. Oh, I don't want to do that, but it's in the cytoplasm. You can see this yellow stuff is supposed to be cytoplasm, and that is glycolysis. And the word glycolysis literally means to break apart glucose. You're lysing or you're breaking or splitting glucose. So glucose is a sugar, C6H12O6. It's a 6-carbon sugar. So it's still a pretty big molecule, right? We want to break it apart into something simpler. So glucose is broken apart into something called pyruvic acid, and that is what glycolysis does. So if you're on your little sheet and it says glycolysis to break apart glucose, you may want to add that glycolysis is broken apart to make pyruvic acid, and this happens in the cytoplasm. Now, this also takes a certain amount of energy. So with glycolysis, you actually need to have some energy to make this reaction happen. So it's broken apart to make pyruvate. A bit of activation energy is needed to start the process. So 2 ATP are used, and then they lose energy and become ADP. When glucose molecule breaks and reforms, the bonds release a small amount of energy, allowing for 4 new ATPs to be made. And 4 high energy electrons are released. They are very high energy and are passed on to NAD plus to form 2 NADH to carry high energy electrons to other parts of the cell. So again, lots of complex words and symbols in there. Essentially, it takes 2 ATP to make 4. So 2 of the 4 that are made are recycled back into glycolysis to kind of get it started again. So only a net of 2 ATP additional, like you have 2 that you don't need, are made in glycolysis. The other 2 are used to start glycolysis over again. And you also create 2 other energy carriers, which are NADH. I honestly don't care if you ever remember the NADHs or the NAD pluses or the NADPHs. ATP is our jam here, right? We want to make ATP. So remember that much. And then you're also talking, these other guys they're called energy carriers. They carry electrons generally, and they move them around to different parts of the cell to be used by different processes. So that's glycolysis, breaking apart a 6-carbon sugar to make a 3-carbon pyruvate molecule. And it takes 2 ATP to make 4. And then 2 of them are recycled back into glycolysis to start over again. And 2 of them are used by the body. We can use those 2 ATP to do other things. The next step is the Krebs cycle. So the Krebs cycle is where we further break down that sugar. So now we have the pyruvate. And there are 2 pyruvates for every 1 glucose. So 2 pyruvates for every 1 glucose. These go into the mitochondria. So the 2 pyruvic acid molecules go into the mitochondria. And then they are broken down further in multiple steps to eventually make some more energy carriers. This is very complicated. Sometimes the Krebs cycle is called the citric acid cycle, by the way. Do not memorize this. I had to memorize this in college. I do not even know it now. So unless you were to become a molecular biologist, you don't really need to know the Krebs cycle by heart. So I'm not going to expect you to memorize this. I want you to realize that pyruvate, there are 2 for every 1 glucose, come into the Krebs cycle, and they go through multiple steps of breaking down, and they release carbon dioxide. So carbon dioxide is released here. And what ends up coming from this is some other energy carriers. So we end up making some ATP as well, but we also make 2 more energy carriers, NADH and FADH2. Okay, so in our Krebs cycle, I'm going to go back to it for a moment here. The Krebs cycle total, this is a highlight because I want you to put this one, is 2 ATPs. It makes 2 ATPs, 6 NADHs, and 2 FADH2s. Again, if you don't remember that they're called NADH or FADH2, that's fine. Just note that they're also making other molecules that carry, these are electron carriers. So they're like molecules, their sole purpose is just to hold on to a bunch of electrons. And so they take those electrons someplace else, and they're going to be used in the next step of aerobic respiration. So again, you have glycolysis where glucose is broken down into 2 pyruvate. 2 ATP are made, actually 4 are made, but 2 are used to start glycolysis over again, and 2 are used by your body. And then the Krebs cycle where 2 more ATP are made, and then a bunch of these energy carriers are made. By the way, carbon dioxide is released here, so when you breathe out, that's the carbon dioxide that has been generated in the Krebs cycle. And then you have the electron transport chain, and this occurs on the membrane, the inner membrane of the mitochondria. So if I look at this picture, you've got this mitochondrial matrix in here, but you also have this inner membrane right here. So the inner membrane is where we see the electron transport chain happening. In the electron transport chain, you actually have little proteins that are embedded inside that inner membrane, and their job is to shuttle those electrons. They pass them on kind of like hot potato. They're just like passing the electron to the next one. And they're also pumping hydrogen ions into the space, the intermembrane, the space here in between the 2 membranes. Okay? So they're passing those electrons on, and those electrons are used to make water. And they're also pumping some hydrogens inside to create a gradient again. We've seen this before in photosynthesis. So you have a hydrogen ion gradient. You have more hydrogens inside than you have outside. This is the mitochondrial matrix. This is inside that membrane space. And since there are more of hydrogens here, it's unstable. So the hydrogens want to get out of there. The only way they can get out is through this fancy protein called ATP synthase. It's an enzyme. So hydrogen gets pumped out through ATP synthase, and it is used, that energy is used to create ATP from ADP. So adenosine diphosphate has 2 phosphate groups. They attach 1 additional phosphate group to it, which results in adenosine triphosphate with 3 phosphate groups. So that's way simpler than the lesson gave you, even though I know it's still complex. But let's go through it again. So you've got your your glycolysis, where glucose is split apart. That's the point of glycolysis. It happens in the cytoplasm. Glucose is split into two of those pyruvates, and a little bit of ATP is made. Four total, but two net ATP are made. Then those two pyruvates go into the mitochondria, and at this point, they are in the mitochondrial matrix, and they are shuttled through a whole bunch of different stages, which result in the release of carbon dioxide. CO2 is given off, and two more ATP, and several of these electron carriers are made. Then those electron carriers, they go to the mitochondrial membrane, and they are shuttled. They pass those electrons from protein to protein, and it results in two different things. Basically, the energy from the electrons is used to make water, and some hydrogens are pushed across inside this space to create a gradient that is then used to make ATP. So two ATP are made in glycolysis, two ATP are made in the Krebs cycle, and 32 ATP are made in the electron transport chain. So lots and lots of ATP is made in this part. This is the most energy producing part of all of aerobic respiration, but it can't happen unless the other two phases happen first. So one glucose molecule yields a total of 36 ATP. So rewatch that if you need to. Hopefully that makes sense. If you're still foggy on this or confused, please reach out to me. We can meet individually and go through this on a one-on-one basis. Again, contact me via webmail, or my extension is 2204. Have a great day.